Hi, hello everyone. Welcome back to my YouTube channel. This is Soumya. This video is on photosynthesis. Especially, we are discussing the concept that is mechanism of light phase. In that, it has 7 steps. And in the previous video, we have been completed photolysis of water, emulsion enhancement effect, and configuration of electron transport. In this video, we are going to discuss molecular configuration and orientation of supramolecular complexes, domains, and assemblies. Okay, and then mechanism of non-cyclic electron transport. So by knowing the molecular configuration and orientation of the supramolecular complexes, then only you can able to study the non-cyclic electron transport, because this gives the idea about how the molecules or the domains or the complexes have been arranged on the thylakoid membrane. So where they are located, so how the electrons are moving from one complex to another complex. So if you know that their configuration and orientation and the site where they are located, if we are thorough in that, then it is able to understand the mechanism of NET very easily. Now, without making any delay, let's begin with this molecular configuration and of supramolecular complexes. First one, in that PS2. So in the previous video, I have been telling that there are two systems, that is photosystem 1 and photosystem 2. So first we will discuss the photosystem 2 here. This photosystem 2, so as you can see in the diagrams, for make you to understand here, you can see in the diagram I have mentioned that there are two systems, that is photosystem 2 and photosystem 1, and we have the complex called as cytochrome B6F. Okay, now I am explaining this complex, what are the molecules are present in that, how they are arranged. Okay, all those things we are learning in detail now. So this PS2 is a transmembrane assembly of 25 polypeptide chain with a molecular mass more than 1000 kilodalton. So this PS2 is a transmembrane assembly of 25 polypeptide chain. Okay, the polypeptide chain is it has a 25 polypeptide chain with a molecular mass is 1000 kilodalton, and PS2 is located on stacked and or appressed membranes. So it is located in stacked and appressed membrane, and PS2 is the system that absorbs red light of shorter wavelength, as you can see in the diagram which I have drawn. So this PS2, we are talking about this PS2. This PS2 absorbs the wavelength of shorter wavelength, that is P680 nanometer. Okay, this PS2 observes a shorter wavelength P680 nanometer. And PS2 comprises three major components, that is LHC2 and core and OEC or WOC. This is OEC, oxygen evolving complex or water oxidizing complex. Okay, LHC means light harvesting complex 2. So it is a PS2, so light harvesting complex 2. Okay, now we will learn one by one here. The first one, LHC2. LHC2, it is a 26 kilodalton transmembrane protein. It is a major external antenna system of PS2. Okay, so this we are talking about this LHC2. This is the light harvesting complex 2. This is the one which actually absorbs here 680 nanometer of photon. Photon means pocket energy molecule. So that is about 680 nanometer of wavelength, which has been absorbing from the sunlight, and this energy molecule is first absorbed by the antenna pigment, that is LHC, light harvesting complex 2. Okay, and then this one is providing that energy to P68 to the photosystem 2. It is a major external antenna system of PS2 and contains 7 chlorophyll A and 6 chlorophyll B and 2 keratin. What is the function? This is important. What is the function of the LHC2? The function is that this absorbs radiant energy and transfers it to reaction center in the core by RET, means resonance energy transfer. This one is going to absorb this radiant energy and it is going to transfer this radiant energy to the reaction center. This is the reaction center, that is P680. Is the reaction center. It is transferring that energy to the P680. Okay, that is the main function here. Next, moving on to the next one, that is second one, core. We will look at what is this core. Core contains two major protein units, that is D1 and D2. And now we are talking about this. The core in the PS2 is the D1 and D2, which are of 32 kilodalton each. And additionally, other proteins called as CP43 and CP47 are also found. Okay, and D1 are encoded by PSBA genes. This one is important. So D1 is coded by a PSBA1 gene, D2 are encoded by PSBD gene. Okay, and it contains a pair of special chlorophyll A molecules called as P68. Okay, and it contains a special chlorophyll molecule called as P68, that is the reaction center. Okay, and this P68, it is a pigment, as I just now mentioned, and it wavelength of absorption of maximum is 680. Okay, and this P680 is the reaction center of the PS2. This is the important, the reaction center of the photosystem 2 is P680. Now, and also you can see that a pair of pheophytin is found. You can see the pheophytin. So this is the pheophytin. A pair of pheophytin is also present, and pheophytins are magnesium lacking and colorless chlorophyll molecules. Okay, and this pheophytin is a colorless magnesium lacking chlorophyll. It instead contains two protons. What it contains? This pheophytin instead it contains two protons. An electronic site called as QA is found on D2. So on D2 you can find the QA. Okay, you can see here, right? So on D2 on DT on D2 you can see the QA. Okay, this is the QA. You can see that on D2 side. And a permanently found immobile electron carrier called PQ, that is plastoquinone, is QA. So this QA is, what is this QA? QA is nothing but it is a plastoquinone. It is a electron carrier. Okay, this electron, it is a permanent immobile electron carrier, means it is going to stay here only, it is not going to move anywhere. So it is just passing the electron to the other electron carriers. Now a second electronic site QB is found on D1 towards the stroma, and this QB, the second electron carrier, this is QA and this is QB, it is found towards the D1, towards the stroma side. Okay, and a tyrosine Y residue called Y161, a tyrosine residue. Okay, this is the tyrosine. Y is the symbol of the tyrosine. The tyrosine residue is found on the D1 towards the lumen. So towards the lumen, towards the D1 side, you can find the Y161. Y means it is a tyrosine. Okay, so that's about this total structure of the PS2. And remember, whatever I have told, all the labelings are important while you are why these are important is while you are studying non-cyclic electron transport system. So these, on this only non-cyclic electronic system is going to take place, because it is a, what is this? It is a mechanism, that is we are studying about the mechanism of light phase, right? The light phase is going to take place in thylakoid membrane, or the complexes. These are the molecular complexes which are involved. So all the points which I am saying, everything is important. Moving on to the OEC or WOC. OEC means, as I said before, oxygen evolving complex or water oxidizing complex, and it contains 33 kilodalton, 23 kilodalton and 17 kilodalton, like 3 polypeptide chains. Okay, which are of 17, one is 17 kilodalton, one is 2, another one is 23 kilodalton, another one is 33 kilodalton. Okay, and it also contains, and also OEC contains 4 manganese ions, calcium and chloride. Say about the oxygen evolving complex, it also contains calcium, chloride. Okay, so 4 manganese. Okay, and this manganese and calcium catalyzes photolysis of water. So this point is important. So they may ask about the ions, which ions will aid in the catalyzing the photolysis of water, that is manganese and calcium. Okay, and this releases oxygen, hence the name OEC. OEC means oxygen evolving complexes. Which are the oxygen evolving complex? These are the ones. Okay, and between PS2 and the next cytochrome B6F complex is a mobile electron carrier, that is PQ. So you can see the PQ here, between these two, cytochrome B6F and PS2, you can find the PQ. PQ is a mobile electron transport. It is a mobile, and these two are QA and QB are immobile electron transport carrier. PQ is a mobile electron transport carrier. Mobile means movement. They can move from one place to another place. Okay, carrier PQ, and many copies of PQs are found in the thylakoid membrane pool. So this is the thylakoid membrane, this is the thylakoid membrane pool, right? On this pool you can see PQ, many PQs on the thylakoid membrane. That's about the PS2. Now we will move on to the next one, that is cytochrome B6F complex. So we are going to talk about this complex, that is cytochrome B6F complex. Okay, it is also a transmembrane assembly of 4 kilodalton. Transmembrane means which are located on the membrane. Okay, and it contains polypeptide chains of length 33 kilodalton, and another one is 23 kilodalton, another one is 20 kilodalton, and another one is 17 kilodalton. Okay, Next points here, and where it is found, this cytochrome B6F, it is found on the both stacked and unstacked membrane. So but PS2 is found, where it is found, PS2 is found in stacked or oppressed membrane, but cytochrome B6F is present on both stacked and unstacked membranes. And 33 kilodalton polypeptide chain contains electron carrier called as cytochrome F with a C-type heme. You can see here this cytochrome B6F contains a electron carrier called as cytochrome F. Okay. And 23 kilodalton polypeptide chain contains electron carrier cytochrome B with 2 B-type heme. So here this one cytochrome B, this cytochrome B contains 2 B-type hemes. And 20 kilodalton polypeptide chain and iron sulfur center, that is FeS. Okay, FeS, which is a single electron carrier, and also one more carrier, that is Fe. Fe means iron, S andre sulfur, S means sulfur. This is a single electron carrier which is present in the cytochrome B6F. And there are 2 electron sites, so you can see the 2 electron sites here, that is Q0 and Qi. Q0 is in the lumen, Q0 is in the lumen, and you can see the Qi in the stroma. So you can see the Q0 here in towards this side in the lumen, and you can see the Qi towards the stroma. Okay, and between cytochrome B6F complex and the next PS1 complex, there is a mobile electron carrier called as plastocyanin, as you can see in the diagram. So this is the plastocyanin. It is present between the cytochrome B6F and the photosystem 1. So between them you can see the plastocyanin electrons. These are the plastocyanin electrons. And it is a copper containing water soluble mobile electron, and this one is important, they might ask, that is copper containing water soluble mobile electron carrier is plastocyanin, which is present between the cytochrome B6F complex and the PS1. I'm hoping this cytochrome B6F complex is understood. Now I will move on to the last one, that is PS1. Okay, PS1 is a transmembrane assembly of 15 polypeptide chain with a molecular mass of 800 kilodalton and more. Okay, and this PS1 is located on unstacked or non-oppressed membrane. It is present on unstacked and non-oppressed membrane. One thing you have to observe here is PS2 is present on stacked or oppressed membrane, and cytochrome B6F is present both stacked and unstacked membrane, and PS1 is present on the unstacked or non-oppressed membrane. Okay, you have to remember these 3 complexes where they have located. And here the PS1, and it absorbs a red light of longer wavelength, that is about P700 nanometer. Okay, it's about the longer wavelength of P700 nanometer. Okay, and again it also contains LHC1, that is light harvesting complex 1, and core and other proteins. As you can these parts are similar like that of the PS2. PS2 is also composed of LHC2 core, and then you can see the OEC. OEC means oxygen evolving complex, or you can say water oxidizing complex. So first we will begin with the LHC1. This LHC1 is, this is the, again obviously we know this is the antenna complex formed by hundreds of pigment molecules that captures photons, transfer the harvested light energy to the second component named the reaction center. So again the LHC1, the function of the LHC1 is similar as like that of the LHC2. So these light harvesting complexes is going to absorb the radiant energy of 700 nanometer of photon, okay, energy, and it absorbs this energy and transfers this energy to the reaction center, and in case of PS1 the reaction center is P700. Next we will move on to the core here. So we will study about this core center here of the PS1. The core is composed of 2 main proteins, that is units PSA and PSAA and PSAB. So you can see that there are 2 main centers here, that is PSAA and PSAB. To these 2 main protein units here. And moving on to the next point, the core contains a pair of special chlorophyll A molecules called as P700. P700 is the reaction center of the PS1. Okay. as I just now mentioned that point. A chlorophyll molecule called as A0 is found. Okay, you can see the A0 chlorophyll molecule. This is the one. A0 chlorophyll molecule is found in the core of the PS1, and a vitamin K1 molecule, that is phyloquinone, is found, and it is represented as A1. Okay, this is the A0, and this one A1, this is the phyloquinone. It is a vitamin K1 molecule. It is a vitamin K1 molecule, what it is called as phyloquinone. Next, and 3 4 Fe4S center are found, means 4 iron and 4 sulfur centers are. So this combination has how many have? They have 3. Okay, are found and are represented as Fx or Fesx. So I have not mentioned this name here. You can just represent it as Fx or Fesx, but I have for make you to understand, I have wrote here. So 3 times 1 2 3 times 4 Fe4S means 4 iron and 4 sulfur, 4 iron and 4 sulfur, 4 iron and 4 sulfur. How many times? 3 times. Okay, this is simply, you can write it here as Fx, but just make you to understand, I have wrote the in detail. That's it. And other proteins, what are the other proteins present in the PS1? PSAC is found towards the stroma. As you can see in the diagram here, this is the stroma side, right? This is the stroma side, right? And here you can see the PSAC, okay, found towards the stroma and contain Fa, and it contains a Fa, this one, it contains a Fa, and it also contains the Fb. Okay, this is the nothing but the iron sulfur complexes. Okay, and also it contains a PSAD and PSAE attached to an electron carrier called ferredoxin to the to photosystem 1. So these PSD and PSE, these two are attached to the ferredoxin. These two are attached to the ferredoxin electron transport. Okay, are attached to the ferredoxin electron transport molecule. And this ferredoxin is a non-heme iron sulfur center containing protein. So this is the non-heme iron containing center of protein, and that's about the molecular configuration of supramolecular complexes. I'm happy to teach you all. Thanks for watching.